So let's start with the latitudinal biodiversity gradient. This has been described in rather grandiose terms as the dominant macroevolutionary pattern, or the first order macroevolutionary pattern on Earth today. What we're looking at here is just one example of the general modern day pattern. So this is a graph for the biodiversity, let's say species richness of extant birds plotted against latitude. And so what you can see is a nice big peak close to the equator, high biodiversity in the tropics, and then it tails off as we get into higher and higher latitudes. Now it turns out that this is a pattern that is repeated over and over again across many different extant organisms, not just birds, but other vertebrates, not just vertebrates, but invertebrates. We see it on land, we see it in marine forms, we see it in plants, we see it in microorganisms. So this is a very strong macroevolutionary phenomenon that biologists studying extant organisms are extremely interested in. Now, as Wolfgang and various other people have pointed out today, as paleontologists, we have some very nice data in the fossil record if we want to try to understand where these patterns come from, how they possibly change through time, what factors affect them, and so on. So there have been numerous studies in the fossil record of latitudinal biodiversity gradients, or LBGs, as I will call them, because I'm going to be using that phrase probably about 200 times in the talk. This is just one example from Palaeozoic brachiopods. You can see their distribution, again, plotted against latitude, with something that looks suspiciously like a peak in diversity, basically close to the equator. So it's been suggested from a very large number of studies that this modern day pattern, where we see a peak close to the equator, actually is essentially an ancient pattern. It's possible that this was set up as early as 500 million years ago and has persisted pretty much ever since through to the present day. Now, of course, this can be perturbed by various events. There's some evidence, for example, that climate change in the late Paleozoic didn't change the overall sort of pattern here in terms of having an equatorial peak in brachiopods, but it did change the slope of the gradient. So there seems to be potentially some kind of relationship between the slopes of these patterns and various climatic gradients. Okay, well, what causes the LBG, or the modern type LBG? Well, at last count, there were something like over 100 different explanations for how these patterns come about. Now, of course, these do not have to be mutually exclusive, and we may be able to combine them together in various ways. But we can group these explanations into three broad categories: historical, climatic, and geographic. Because of time, I'm not going to say very much about geography. And with regard to history, what I mean by that really is the legacy of previous evolutionary events, perhaps a major diversification or perhaps a major extinction event. So, for example, the LBGs for modern day ants in North America are not symmetrical with regard to those in the southern hemisphere in South America, and that possibly reflects an extinction event amongst North American ants perhaps 30, 40 million years ago, and they're still kind of recovering from that perhaps. But let's have a look at climate in more detail. If you read the modern macroecological literature, for example, there's a study by Field et al. In 2009 that was a meta-analysis of over 400 analyses of extant organisms. There's a very strong view that climate is the control on the modern day LBG. And you can see a couple of example factors here. One is, of course, we have greater insulation near the equator. So you've got more energy coming into the system, potentially more productivity. That allows species to have to maintain larger population sizes. And what that might mean is that perhaps they have some buffering against extinction. So maybe species in the tropics are more long-lived. This is the idea of the tropics as museum hypothesis. And so biodiversity might build up in the tropics because things die out more slowly. Combined with that, you may have a factor such as decreased seasonality in the tropics. And that means that you've got nicer, more stable conditions. Species can become more specialized, so we get this idea of increased stenotypy. So you're much more focused on doing one particular thing in one particular place, and that allows the tropics to maintain a higher level of biodiversity. You can cram more species into the same area. So those two factors together potentially could combine to set up the modern type LBG. However, there are some exceptions, even today. So a couple of the modern amphibian groups, like the tree frogs and salamanders, and also the albatrosses, they do not show the modern type pattern. So that raises another set of questions. We have one major pattern that we'd like to explain. Where does it come from? What controls it? But it's also interesting to think about why we get exceptions to this pattern. Okay, well, that's right, yeah, okay. Exceptions to the pattern. We had an idea a few years ago that it would be interesting to look at LBGs in the terrestrial fossil record, because the vast majority of papers had focused on the marine fossil record. And it's one of those ideas that you come up with it and several other people come up with it at exactly the same time. And so we now have 3 different studies of ancient terrestrial LBGs, which basically take the subject a little bit further, because as Wolfgang was pointing out, it's important to consider aspects of sampling. The peak you see in diversity in the fossil record with regard to latitude, if you just take the raw numbers, might simply reflect the amount of rock you have available, or particular places that paleontologists like to go and visit to collect fossils. So all of these studies here, one by dinosaurs by myself and my colleagues that I'll be going into more detail, one by Archibald et al. On Cenozoic insects, and one by Rose et al. On Cenozoic mammals, all take sampling into account. And what they found is potentially a rather interesting and unexpected pattern in the terrestrial world. So with the Archibald et al. Study on insects, for example, what they found was that the insect fossil record doesn't show a modern type LBG until we get to some point in the Eocene or slightly afterwards. So before that, insects didn't conform to the general pattern. In the Rose et al. Study on mammals, what they found was that if you, for example, look at a Palaeocene record of North American mammals, again, they show no latitudinal gradient as we go from lower latitudes to higher ones. So again, it's not fitting the modern pattern. Things seem to be different on land prior to the Eocene or prior to the Oligocene. And now what I want to do is basically focus on that in more detail by looking at our analysis on dinosaurs. So we've been working within the PBDB. This is a great set of resources for this type of analysis, particularly for the dinosaurs, which have received a great deal of careful taxonomic attention. This is one of the more completely represented terrestrial groups in the PBDB. This dataset is already, the one we've analysed is already a little bit out of date because things move on very quickly, but we were able to work with about 740 dinosaur genera and about 3,500 occurrences for their 160 million year Mesozoic fossil record. So you can see that sampling is rather sparse. But we were able to represent all of the dinosaur groups, including Mesozoic birds, and we were able to get dinosaurs from a wide variety of different latitudinal bins. We have some dinosaurs very close to the equator. We also have some dinosaurs from polar regions up to about 80 degrees north and south of the equator. So they look like quite a good data set for investigating the Mesozoic. And there had been no study of terrestrial LBGs, believe it or not, for Mesozoic organisms that had taken sampling into account. Not that I'm aware of, anyway. Okay, well, sampling is important. So this is a set of curves that relates to diversity and sampling through time. So you can see Mesozoic stages on the x-axis there. The black line relates to the number of genera you can see in the raw fossil record. The red dashed line and the blue lines represent ways of estimating sampling. Basically, DBFs refers to geological units, numbers of formations where you can find dinosaur material, and DBCs relates to the number of collections made by paleontologists of dinosaur material. And you can see that those three curves, with a few exceptions, basically track each other through the Mesozoic. That's just a kind of qualitative eyeballing. However, you can apply some stats to this, you can detrend your data, you can look for a correlation statistically, and you find that yes, actually, these three curves do correlate with each other. And in fact, dinosaur diversity correlates with other measures of sampling, such as rock outcrop area as well. So sampling is going to be a key component in the dinosaur fossil record, and therefore when we look at it with latitude rather than with time, we need to take that into account. So we threw a wide variety of different techniques at this problem for a couple of reasons. There are a wide variety of ways of dealing with issues of sampling in the fossil record. Wolfgang gave a very nice illustration of one of those, the sort of rarefaction subsampling approach, and we applied that. Recently, John Elroy has produced a modified version, quite a heavily modified version, called shareholder quorum subsampling, or SQS, which takes into account the relative abundance or frequencies of occurrences of various species, because it's been claimed that if you get a very heterogeneous fossil record where some of your taxa are represented by hundreds of individuals and some of your taxa are represented just by singletons, that can cause a problem for standard subsampling. So we applied that as well. We also applied the modelling approach, the residuals approach proposed by Smith and McGowan in 2007, and it's been used in a variety of studies of mainly vertebrates since then, in which you take a sampling proxy like rock outcrop area and you compare that with your raw diversity, and you essentially produce a model that says, let's imagine that sampling perfectly predicts and controls the amount of diversity we see in the fossil record. That gives you a prediction as what you would expect to see if everything is controlled by sampling. You can then compare that with your real data and see where you get discrepancies or residuals. And on the bottom right here, that's the diversity curve for dinosaurs. These are residual diversities when you've tried to remove the effects of sampling in this way. We also used some multiple regression techniques to try to look at what was controlling dinosaur latitudinal diversity. We could use controlling or explanatory parameters such as various sampling measures, but we could also put in things like a land area to see what might be explaining the distribution of dinosaurs with latitude. Okay, well, basically what we found was that dinosaur diversity doesn't conform to the general pattern, the general modern day pattern. It's another example where prior to the Eocene, in this case the Mesozoic, but particularly with regard to the Cretaceous, we could not see any equatorial peak in dinosaur distribution. So this was kind of interesting to us because it sort of reinforced this idea that things were different on land. So sorry, this is the residual diversity, and what you can see there is the dashed lines, horizontal lines, represent effectively sort of 95% confidence limit. So you can see that most of our latitudinal diversity is actually varying within that. Therefore, we've got to be very cautious about how we interpret that because it's not necessarily statistically significant, but there are some peaks and troughs that touch those lines. And the peaks we get, the major peaks in dinosaur diversity are around about 50 to 60 degrees north and south of the equator. Now, just want to go back to this one, explain a little more about what's going on. This is the result from rarefaction, from subsampling, and what you find is that we get lower diversity in our 0 to 30 degree bins and higher diversity in our 30 to 60 degree bins. So again, these two methods, which work in a very different way, are pointing to the same issue, that dinosaur diversity during the Mesozoic was higher at mid-latitudes and lower at equatorial or tropical latitudes. But notice that, as I mentioned, we've got to be very careful about the error margins here, and on our rarefaction analyses, we had to take very, very large latitudinal bins in order to get this result. I'll come back to that later when I talk a little bit more about big paleo and databases. We got another interesting result. This time what we did was we split our dinosaurs into separate clades, two major clades in this case, the two major plant-eating groups, the Ornithischian dinosaurs, you can see a Triceratops on the left there, and the sauropod dinosaurs, you can see a nice sauropod, long-necked herbivore on the right. The sauropods are the largest terrestrial animals of all time. It can take about 5 people just to lift up one of their thigh bones. That, until today, was my idea of big paleo. But what we've done here is basically reconstruct their latitudinal distributions for these two different herbivorous groups of dinosaur for the late Cretaceous. And what we found was some interesting separation, sort of ecological, geographic, latitudinal separation between these two herbivorous groups. Basically, the sauropod dinosaurs tend to be more diverse at lower latitudes, round about, let's say, 30 to 40 degrees, whereas the Ornithischian dinosaurs tend to be more diverse at high latitudes of 50 to 60 degrees. Also, the sauropods tend to be more diverse in the southern hemisphere, and the Ornithischians more diverse in the northern hemisphere. So the discrepancies in these peaks might reflect some ecological separation. Maybe there are certain plant groups, or maybe aspects of physiology of these animals that mean that sauropods were more successful at lower latitudes and Ornithischians more successful at higher latitudes. But the difference between the north and the south is potentially a legacy of perhaps some kind of mid-Cretaceous extinction in sauropods in the northern hemisphere. They seem to suffer quite badly in the northern hemisphere, in particular during the mid-Cretaceous, and so perhaps they hadn't really recovered from that. So what we might be seeing here is essentially a combination of, sort of ecological factors and these sort of historical factors that I mentioned earlier. Okay, well, getting to the slightly bigger picture and fitting it into the more modern context, what we find from the Rose et al. Study, the Archibald study, and our own study is that prior to the Eocene, on land, when you take sampling into account, you tend to see peaks in diversity in the temperate zone or depressed diversity in the tropical zones. Now, why might this be? Well, it seems strange that should occur if everything is controlled by climate. But we'll come to climate in a moment. One possibility is that, think about the Cretaceous and perhaps into the early Cenozoic. If we have a greenhouse world, very warm conditions, reduced seasonality, perhaps reduced or shallower thermal gradients as you go from the equator to the poles, that might simply cause a shallower gradient for the LBG. You might still have a peak in the equator, and you might just have more diversity in the temperate zone. That wouldn't fit our pattern. So there's something else going on here. Why would diversity be lower during the tropical zones? Well, we come up with a working hypothesis. I stress this is a working hypothesis. Still need to do a lot of work on this. But the idea would be that perhaps during particularly warm periods, because of shallower gradients or reduced seasonality, other factors aside from climate, which were previously overwhelmed or suppressed, might have a stronger influence. And one of the ones we tested using our multiple regression models was the idea of land area. So we took paleogeographic maps, we used those to estimate the amount of land area per latitudinal bin, and we found that there is indeed a strong correlation between the amount of land area in the Cretaceous and the amount of diversity you see amongst the dinosaurs. So the idea is that if you remove or reduce the climatic control, then perhaps another factor like land area starts to have an effect. And of course, dinosaurs are a completely terrestrial group. They didn't produce any marine forms. During much of the Cretaceous, much of the tropical zones were ocean, so why would we expect to see many dinosaurs there? So this kind of, I think, fits quite nicely with what we see paleogeographically. Okay, well, I just want to summarize some conclusions before saying something about sort of future work and big paleo and so on. Just to draw it together for you. Dinosaur diversity is pretty much consistently highest in temperate zones throughout much of the Mesozoic, and that perhaps tells us that there's more to explaining this than just climate. It seems unlikely that climate could cause that, or could cause that level of sort of stability over such a long period of time. There must be other factors going on here. But if we reduce the effect of climate, then perhaps other factors like land area start to take over. When you partition your data further into smaller and smaller groups of organisms, you can start to pick up some interesting patterns. And I've already said, you can tease apart the possibility that different herbivorous groups had different latitudinal patterns, and that might reflect their ecology, their physiology, their past evolutionary histories, and so on. But the big story here, I think, is that all the evidence we have so far is that, at least on land, the modern type latitudinal biodiversity gradient may not have started to form until, let's say, around about 35 million years ago, as the world makes a transition from a warmer sort of greenhouse world into a cooler icehouse world. So we don't have to say that climate isn't important in determining terrestrial LBGs. It is. And it certainly seems to be at the current time. But that doesn't necessarily hold true throughout all of geological history. And it may be that we have to be a little more sophisticated in how we sort of set up these rules about various factors controlling biodiversity. It may be that different rules operate during a greenhouse world than in an icehouse world. Well, just to sort of look to the future a little bit and say something about big paleo. This work on dinosaurs was actually sort of a pilot study for a grant application to the Leverhulme, and I'm glad to say they gave us the money. So what we're trying to do now is build a bigger and more comprehensive data set for all Cretaceous terrestrial tetrapods. It'd be nice to put all vertebrates in, but we'll at least deal with the tetrapods. We've been working within the PBDB. My colleagues, Roger Benson and Phil Mannion, just during the first 9 months of this project, I think have probably added somewhere in the region of 10 to 15% to the PBDB with regard to Cretaceous terrestrial tetrapods. So we've still got some time to go on this, and I think we can really improve the quality of this data set. The thing about the PBDB is it can be quite patchy in its coverage, so the dinosaurs were already in very, very good shape. Lots of attention being focused on those. But other groups, such as the mammals and the lizards and snakes and so on, were relatively poor. And there are still some groups that need a lot of attention. I've listed some there. Freshwater fish, this amphibians, turtles and so on. We need to focus much more on those if we're going to have a nice comprehensive data set. Also, it's not just a case of putting these things into the PBDB and then printing out a data set and then using it. You have to sift your data quite carefully. And I think it's very important that with these big sort of macroevolutionary, macroecological studies that are going to come out of these internationally set up data sets, that we work with taxonomic experts so we can carefully sift the data. Because, as we've already mentioned a few times, there are issues about, data quality, how many synonyms are there, are the dates for the formations that these animals come from correct, etc., etc. That said, once we have our data set, I think we'll have enough data to be able to answer some more interesting questions. And in particular, as I mentioned before, at the moment, for example, in our subsampling studies, we had to take 30-degree wide latitudinal bins. Now, we could get away with much smaller bins for residuals, but for rarefaction, we needed very wide bins. And that, I think, is a concern. We'd like greater temporal and spatial resolution. So with a bigger data set, you can then slice it up more finely, and hopefully you can do things with that. Secondly, at the moment, we're really just thinking along the lines of, what were tetrapods as a whole doing during the Cretaceous? But that's a very coarse level to be working at. What we'd like to do is start asking questions about, do freshwater groups do something different from terrestrial groups? How does body size affect their latitudinal distribution, and so on? And I think if we can do that, then we can start to tease apart some of the biological factors from the climatic factors that possibly control LBGs. So just some acknowledgements. I'd like to thank the Leave Human course who are funding this. And I'd like to thank those people below that for their data and helpful discussion. I should particularly mention Anthony Goswami, who is a co-I on the Leave Human grant and is hopefully going to be making sure that at least the mammal part of our data set is accurate. Thank you very much.